right, good afternoon, everybody. It's a real pleasure for me to welcome Linden Parks, whose work on an array of issues regarding functional connectivity and biological psychiatry are things that I've been following the last little while. We've had the chance to interact a few times, I think, through HPM. So it's nice to see you be able to come here and provide your talk to us today. So Dr. Parks is a postdoctoral research fellow at the University of Pennsylvania in the Department of Bioengineering. The central goal of Linden's research is to better understand mental illness through brain research, to better understand the brain behavior and brain symptom relationships that are relevant to mental illness, and being cognizant of their biological underpinnings, reveal their predictive capacity, translate our predictive insights into prognostic impact, and uncover developmental pathways that give rise to brain network dysfunction. His work spans these domains and is highly cross-disciplinary. And I would add on another note that he's written a lot of impactful papers, both methodologically and in terms of biological psychiatry, that are definitely worth delving into and are required reading for many people in my lab. So with that, Linden, thanks so much for being here today. And I'll turn over the floor to you. Dr. Parks also was offered to take clarifying questions during the talk. So either you can chat them in or raise your hand or interrupt. For maybe deeper, broader discussion questions, if you could save them until the end, that'd be great. So it's all yours.
Cool. Thank you, Mala, for the invite and for the kind introduction. I'm really pleased to be here to talk about some of my postdoc work. All right, so today I'm going to deliver a talk about some of the work I've been doing during the last few years of my postdoc under this title here. Broadly, this talk will kind of be about how I use network models to study individual differences in the connectome and how I kind of use tools from network science to predict phenotypes of interest to me, like sort of spanning across psychopathology and developmental processes and cognition. Throughout my career, as Mala said, I've been fortunate to do a really kind of broad range of research focusing on different topics, including some method stuff. But for me, like the thread through my work that kind of ties it all together has always been a focus on studying individual differences. And I've always kind of been very interested in how individual differences in the human brain map onto individual differences in mental health symptoms in particular. So that's what we're going to talk a lot about today. So in the context of psychiatry research, the kind of epitome of this approach, this individual differences approach, is of course the field of biomarker detection, which is simply the idea of trying to seek out objective measures of a person's biology that are indicative of their kind of disease status. For a very long time, of course, the primary approach to doing this, to detecting biomarkers, has been the case control design, which involves finding group level effects in the brain. This design assumes that patients are kind of cleanly separable in terms of their symptoms as well as their underlying biology, and that this separability should aid us as sort of translational scientists in differential diagnosis. Now, while we've certainly learnt a lot, I think, about the link between disorders in the brain, as a field, we've sort of slowly come to terms with the fact that this assumption of separability isn't really borne out in the data. Not at the level of symptoms, nor at the level of the biology. And so, when we consider how we diagnose patients, this lack of separability is probably not so surprising. We know that patients can be diagnosed with a certain disorder in many, many ways, which leads to kind of groups of patients that have very high levels of symptom heterogeneity, despite the fact that they're all assigned the same label. In turn, it's not so surprising then, of course, that groups of patients who look dissimilar clinically, also exhibit biological heterogeneity. And indeed, there's like a lot of work now in the field of psychiatric research that sort of supports this notion that case control designs are kind of limited in this issue of heterogeneity at multiple levels. So this suggests to me, and of course many others in the field, that our reality as researchers is probably much more dimensional than it is categorical. And a great example of this is something like the psychosis spectrum, which shows us that psychosis symptoms are not exclusively present in disorders like schizophrenia, but they're also present to sort of varying extents in the broader population, and perhaps they're even present a little bit in most or all of us. In turn, this kind of dimensional perspective says that, while maybe the very extreme expression of psychosis symptoms might underpin disorders like schizophrenia, studying that kind of full spectrum, may not own, studying that full spectrum and the link of that spectrum to the brain, might not only improve our understanding of pathophysiology, but hopefully would also improve our sort of capacity to be prognostic and to make treatment decisions. Another layer to this is that this dimensional perspective doesn't occur in a vacuum, but rather sort of any specific dimension, like the psychosis spectrum I mentioned, is probably part of a broader kind of transdiagnostic space, which kind of assumes that, multiple symptom dimensions, as well as dimensions of cognition and other developmental processes, sort of likely all interact and are likely underpinned by sort of multiple similarly interacting biological signatures. And so this is kind of where much of my research sits, or that's how I like to see it. And so today I'm going to present some results from a couple of projects from my postdoc that examine to what extent, like as someone as a network neuroscientist, can kind of help pass out and explain sort of variance in this transdiagnostic space, both in kind of patient data and population data. So what does it mean to say that I'm a network neuroscientist? I'm just going to provide a little primer for those who perhaps aren't in the field. So network science is really just the formal study of complex networks and complex systems. At face value, these networks are like very intuitive things, and you'll probably be no doubt aware of many of them in your own world. For example, networks connect us socially, the most ubiquitous example of which is social media and Twitter, which allows us academics to kind of argue with one another online. Networks help us to navigate. If we know the connections that make cities, we can plot paths that help us sort of traverse the globe through a series of flights. And additionally, networks can be at different scales as well. So in this transport network examples of networks that are kind of more local scale than the entire globe are networks of sort of road systems within countries. Now here, like even to the naked eye, it becomes very clear that there are kind of potentially very interesting differences between these pairs of networks. They have very different kind of distributions of locations and connections between those locations, different paths. And one of them, Australia on the right, is clearly much sparser than the United States on the left. And finally, and the example I want you thinking most about today, of course, is that our brains are very easily thought of as networks too, which we in the field often refer to as the human connectome. So casting the brain as a network in this way kind of really opens up lots of cool opportunities to study the brain. We can kind of ask questions about how the brain communicates, how it integrates information, what are the sort of properties that support it doing that. And we can kind of examine how the kind of complex web of connections changes in individuals who experience mental health. And particularly equipped with the language of network neuroscience, we can also kind of try to hopefully start to unravel how complex dynamical patterns emerge from the network. So this cool video here is from the zebrafish brain, and it shows sort of waves of spiking activity traveling throughout the zebrafish brain. So one of my goals today is to convince you that while maybe we can't study dynamics quite as precisely as our colleagues who study the zebrafish, we can leverage network neuroscience together with with neuroimaging to study certain aspects of dynamics in the human connectome. All right, so as an extension of this quick primer for those unfamiliar with some network science terms, within the human connectome, we kind of tend to have two basic key components or terms that we use to describe those components. First, we think of brain regions as nodes of our system, and we define these typically according to some parcellation of the brain, of which, of course, there are heaps to choose from. And second, we think of the connections between those nodes as edges of our network. And it's important to note that edges in particular can be defined in many, many different ways, and how they're defined kind of informs and constrains the types of questions that we ask or we can ask when we're studying the human connectome. So for all of the work that I'm going to present today, we're going to consider edges that are defined according to tractography performed on diffusion imaging data. So defining edges this way basically gives us an estimate of the number of white matter, physical white matter pathways that connect pairs of regions. So once we have our, in this case, structural connectome, well, what does it give us access to? Well, the answer from the field is that it gives us access to this thing called topology. So topology simply refers to the idea that the actual relationship amongst the units of the system, the actual kind of higher order pattern of how the nodes are connected by all of their edges, is in itself an important feature to study. And it's an additional feature beyond just looking at the actual sort of weight of a given connection and how that changes. So topology kind of tells us information about, which nodes are more important than others in the network. It tells us how nodes might be grouped together or clustered. It tells us where important, connections might be missing in the network. That if, we were to add, it might improve efficiency. And it tells us information about the sort of pathways that route information throughout the network. And all of these various different features of topology, as well as many that I'm not showing, they each tell us something different and interesting about the connectome. And we as network scientists tend to kind of draw on these features when we're trying to address our research questions. One particular emerging field that sort of leverages this topology and that I'm going to focus on a lot today is called network control theory. So in essence, network control theory involves taking the connectome and kind of layering on top simulated dynamics at every node of the system. I'm illustrating these kind of simulated dynamics here, just using the orange time series. And then we use that model, we ask questions, well, about how the kind of topology of the system supports and constrains the spreading of those dynamics along the connections. And we look at what type of aspects of topology support that spreading. But control theory is also interested in modeling control signals, as the name implies, which I'm illustrating as a separate signal here as the blue time series. These are control signals that we model and then we inject into this now dynamical system, which we then in turn design these control signals, try and see if we can effectively control those spreading dynamics and kind of get them to go where we want them to go on the system. So let's just try to build a little bit of intuition about how this works, because it's a key method for the talk. So in the lab that I'm in Danny Bassett's lab, we've been approaching this with a relatively simple model that follows this equation here. So I'm just going to try and unpack it now to see if I can build some intuition. So this model is a linear dynamical model where the output x dot simply tells us how the activity of brain regions across all the brain regions on the nodes of the system is changing over time. And this change in activity is a function of two factors. The first factor is the internal dynamics, right? So the blue time series, the orange time series I was showing you before. And this is just the idea that the activity at each node kind of diffuses from region to region using a linear model along all of the structural tracks. The second factor is there's any additional control input that we provide and that which can be thought of, and you can kind of think of this control input as either maybe some simulated external stimulation, right? So maybe you're trying to simulate something like TMS, or even you can think of it as like simulating some idea of internal cognitive control. But either way, the general idea to think about it is that it's a signal that we model that is external to the system that we are injecting into the system over time to create changes in brain states. So the model simply simulates basically the evolution of brain states as a function of both the internal diffusion of activity along structural paths on one hand, and the external stimulation that we might inject into the system. All right, so to try to really hammer this point home, let's consider a very simple toy network here of 5 nodes. Each node's activity is sort of low to begin with, as represented in blue. And what we're going to do is we're just going to start by simulating some stimulation at node 3. So when we first stimulate node 3 by injecting some activity, its activity increases. And then the next time step, we'll stop adding a simulated pattern of injected control, and we'll just let the activity diffuse. And from there, it diffuses from nodes 2 to 5 along the structural tracks, increasing their activity, which then propagate to nodes 1 and 4 through the continued connections along those structural paths. And assuming we didn't stimulate any more than that initial burst into node 3, then the activity would just keep diffusing around the system over time, like so. And finally, we end up back where we started. So now this is just a very simple toy example where we only injected a small amount of energy at one time point into one node, and then we just watched the activity move around the system until sort of settling back down to a baseline. So in other words, we only used our second term in our equation to inject some control signal at the start, and then we stopped injecting control, and then we just let the dynamics unfold according to the first term. But in application of these methods to brain networks, we tend to try to design much more interesting control tasks than this in the context of studying the human connectome. So one of the common things that we do with this method is that we define, say, pairs of brain states that are of interest to us as neuroscientists. So just picturing these here, these could be, for example, canonical sort of brain systems. And we try to use this method to define control inputs that move activity from one brain state to another. And then doing that, we can study how the topology of the network made that transition possible. So in this example on the left, we might be interested in finding the types of control signals that we need to move activity from, say, the somatomotor state or the somatomotor system at the beginning to the visual system at the end. But really, we can do this with any pair of brain states that we care about. So when we do something like this, there are broadly kind of two ways we summarize this control information. First, we quantify the amount of energy that it takes our model to complete this state transition. This energy is simply just like a summation of the amount of control that we had to inject into the system at each of its nodes in order to kind of complete that transition between pairs of states. And basically, this gives us an estimate of the amount of kind of effort, if you like, that the model had to go to in order to make our state transition occur. The second is kind of a more general form of output, and we just call those controlability statistics. Instead of, say, estimating the energy associated with a specific state transition, we might be more interested in a brain region's kind of like general capacity to control, like, general in many different types of brain states or different types of transitions. Today, I'm going to focus on a controllability statistic called average controllability, but I just want to note that there are many more that you can choose from. And the intuition I want you to have here is just the idea that a region that has high average controllability is kind of better positioned in the topology of the network in order to kind of orchestrate changes in lots of different brain states. So it's just a generally good controller of dynamics, if you like. And today in my talk and in some of the projects, I'm going to leverage both of these things. We're both going to look at the control energy of specific state transitions and the controllability of brain regions to study individual differences in the connectome. Finally, in addition to layering on models of dynamics on top of the connectome, today I'm also going to try and show you how we can connect our network models with information about the brain's cytoarchitecture, which is the study of the cellular composition of brain regions that is thought to inform structural connectivity and potentially to sort of inform structural topology. So with that context in mind, for the rest of the talk, we're going to discuss a few projects and questions that are kind of all in service of this idea of using network control theory to try and, as I said, explain variance in this transdiagnostic space. And we're going to sort of focus on, for the most part, trying to improve the prediction of psychosis symptoms and to sort of improve our understanding of that link. So we're going to start by asking a very simple question of, does network control theory improve the prediction of psychosis symptoms beyond canonical measures of network topology like the ones I showed at the start of the talk? And then we're going to move into a project looking at whether or not we can use information from cytoarchitecture to kind of constrain the topology and inform brain dynamics. And then finally, we're going to sort of come back full circle to prediction and see if what we've learned by integrating cytoarchitecture actually improves prediction of psychosis even further. But let's start with question 1. So to address this first question, we started by just examining the kind of capacity of this general metric of average controllability that I mentioned earlier to predict psychosis symptoms. And we wanted to compare it against a canonical regional measure of topological importance called strength, which is sort of shown illustrated here on the left in green. Now, a region's strength is simply the sum of the edge weights, so the white matter pathways that connect to that node, where like higher strength just suggests a region that is more connected and therefore kind of maybe more important in the topology of the brain. But there's an important distinction between strength and average control. Strength is limited to only telling us about the local importance of a region to a network topology, because it only measures how directly connected a region is to the rest of the system. Average control, on the other hand, takes advantage of that model of dynamics to kind of access the full breadth of the topology in order to influence or control brain activity across all of the system, not just the nodes that it's directly connected to. So this makes average controlability sort of sensitive to variation in the entire network and its topology, whereas strength is just sensitive to variation in the local connectedness of the node. So what we did in this project was we just extracted strength and average control from every brain region in approximately 1,000 subjects, aged 8 to 22 years. And then we used those two feature matrices shown on the left to basically train a very simple cross-validated out-of-sample regression model to try and predict psychosis symptoms out of sample. And when we do this, we find that average controlability is a significantly better predictor of psychosis symptoms. So these distributions here show error values from our cross-validated model under many, many iterations. So lower here is better. And then in addition to that, we also extended this further to include a broader array of canonical measures of network topology. These three entries on the x-axis correspond to several other measures of regional topology. Simply for time, I'm kind of skipping over the details of explaining these metrics, but the main take home here is that there are lots of different ways in which we can measure topology that we can extract from the connectome and that don't depend on our control model, and that average controlability is the best predictor of psychosis symptoms amongst those competing alternatives. In addition to that, we found that the average controlability was the only region, the only feature to predict beyond chance levels using a permutation test. So this is very exciting to us because it tells us that, at least in our hands, average controlability was really the only one that robustly was able to predict psychosis symptoms out of sample. So this was a really great start, we thought, but we wanted to dig in further and kind of try and build some intuition into how this prediction, improved prediction, was occurring. Oops. So to dig into these results, we first initially turned to the idea of the gradient of functional connectivity, which is pictured on the left here. This gradient probably needs no introduction. It's of course released by Daniel Margulies several years ago, and it provides a sort of putative hierarchical ordering of the cortex that places lower order sensory and motor regions at one end in blue, in my case here, and higher order default mode cortex at the other in yellow. The reason that this is super valuable to us is that it gives us a way of asking how regional controlability varies over the cortical hierarchy. This is important, of course, because we think of the higher order kind of default mode cortex as playing an important role in integrating information over the cortex and kind of routing information between other distributed systems. So this got us thinking that potentially our measure of control was actually serving as a better measure of topological importance of these higher order brain regions in particular, right? Because these are regions that are thought to be important for control, and we're kind of claiming to model that control. If that's true, we reasoned that this might be where we're getting better prediction for average control. So happily, we found some results that sort of support exactly that. Starting on the left subplot here, every point in this scatter plot is a brain region, and the x-axis is that region's position on that hierarchy of functional connectivity, with the default mode cortex being on the right-hand side. For each region, we simply just took the values of regional strength and control across all the subjects, then just correlated them together. So every point here, the y-axis is like the cross-subject correlation between the two features. So a correlation of 1, or a correlation approaching 1, would mean that strength and control are sort of redundant measures of individual differences across subjects. And what we observe is that this correlation actually kind of decreases as regions traverse up the cortical hierarchy. showing very clearly that average control and strength exhibit increasing amounts of unique variance at the top end of the cortical hierarchy, right? Because that correlation is reducing. This is important because while, as I mentioned before, while strength only captures local topology, as I said, because it's only sensitive to direct connections, average controlability kind of captures the full topology or variation within the full topology. So what this tells us is that this divergence between our two features tells us that network control captures something different about the topological importance of these higher order regions. But most importantly, if we now look at these middle and right subplots here, what we also found was that this divergence seems to translate into prediction performance that improves selectively for average control as we move up the gradient. So in these plots, what I'm showing is I repeated our cross-validated prediction model just using subsets of brain regions traversing up that hierarchy. And when we do this for each feature, we find that prediction gets better for average control as we move up the gradient, but it remains uniform for strength. So again, this tells us that average controllability's kind of capacity to access that full topology and to kind of really leverage it from those default mode regions is what drives its superior predictive performance compared to strength. Okay, so we thought this was a wonderful start. Collectively, we've kind of said, well, with this, our results point to this idea that the psychosis spectrum is associated with kind of disordered control of dynamics that sort of stem from the default mode cortex, and that our model, our simple model of control theory, kind of gives us a clear advantage in studying that dysfunction. So we've addressed that first question, right? Does network control theory offer us something better in terms of translational neuroscience over and above kind of standard measures of topology? But you know, we also showed, and we showed that this vary in the functional hierarchy. Now, while the functional hierarchy is nice and all, it's a long way from what I proposed at the start of the talk, which is around grounding our models in cytoarchitecture. And as such, for this next project, I want to turn our attention to examining whether and how cytoarchitecture constrains the dynamics of our system and kind of affects the resulting model outputs. And then, as I said in the final section of the talk, we'll kind of go back to this idea of saying, well, can we integrate cytoarchitecture and can we get even better prediction out of our predictive models? So there's a really long history of evidence from neuroanatomy that tells us that brain regions profiles of cytoarchitecture informs how pairs of regions are structurally connected. So as I touched on briefly earlier, cytoarchitecture is the study of the kind of cellular composition of brain tissue. So in the cortex, it involves studying, the makeup of different layers within the cortical sheet, which is sort of pictured here on the left. It's interested in how many layers, examining how many layers there are in different regions, how clearly defined these layers are, and the density of different cell types within each of these layers. But most importantly, this line of research tells us that the interregional similarity in cytoarchitecture profiles plays a really key role in determining the pattern of feed-forward and feedback projections that kind of connect region pairs. And it makes predictions about the asymmetries in these projections between pairs of regions. So in short, cytoarchitecture tells us something about the nature of structural connectivity between regions. And a very exciting recent advance for us has been the release of the ultra-high resolution histological atlas of the human brain, which is called the Big Brain Dataset, pictured in the top right-hand corner here, which gives us a look at these profiles of cytoarchitecture in the human brain. And additionally, this atlas has recently been summarized as a gradient of its own right, which is shown down in the bottom right-hand corner here. This is work from Casey Piccola that describes the extent to which pairs of regions have similar or dissimilar cytoarchitecture in the human brain. So I find this really exciting because this map allows me as a network scientist to kind of integrate my network models with a precise map of how regional profiles of cytoarchitecture actually vary across the cortex. And in turn, this kind of opens up lots of cool opportunities to draw links between the topology of the structural connectome that we study and the microscopic features of brain structure that control or sort of govern and predict patterns of extrinsic connectivity. So with that in mind, for this next project, I wanted to use control theory to ask, does cytoarchitecture constrain topology and inform brain dynamics in our model? Specifically, we reasoned that, if cytoarchitecture explains asymmetries in the structural connectivity and the patterns of connectivity between regions and shapes topology, then we might expect that the topology of the network to result in asymmetries in the way dynamics from our model kind of traverse the connectome, and that these asymmetries in dynamics and these differences in dynamics might be explained by this pattern of variation in underlying cytoarchitecture. Then if those asymmetries in dynamics are sort of biologically meaningful, then we reason that they should also link to functional data, which we will measure with ECOG data, and I'll show you in a moment. And we also reason that they should show developmental effects in our developing sample of 8 to 22-year-olds. And I'll explain why that's true in a moment. But first, I just want to quickly go through the approach for this section. So to examine this kind of broad question, what we did was we started by sampling groups of regions that have similar profiles of cytoarchitecture according to Casey-Piccola's gradient. So the gradient of cytoarchitecture is shown here on the left. It's from the Big Brain Atlas. It's just a different color map compared to the one that Casey was using. So basically the idea is if I say sample 2 groups of regions based on different positions on this hierarchy, showing some examples here on the right-hand side, both groups will have internally fairly homogeneous or similar profiles of cytoarchitecture. But those groups, the profiles of cytoarchitecture will differ between the 2 groups. And importantly, they become more different or increasingly dissimilar with kind of greater separation along this hierarchy, right? Like if we were to sample patches of cortex that were very nearby on the gradient, they would have more similar profiles of cortical cytoarchitecture than if we were to sample them at greater distances along this gradient. So what we do is we sort of sample sets of regions from this hierarchy, and we take those groups of regions and we carry them forward into our control framework, and we treat them as brain states. So what this lets us do is it lets us examine transitions between patches of cortex that are located at different positions along this hierarchy. And importantly, here what we're arguing is that we can model in a fairly straightforward way bottom-up and top-down state transitions based on the idea that if we have one patch of cortex on this hierarchy and then another, then the second patch will either be higher or lower than the initial patch, right? And so moving dynamics, or trying to use our model to control dynamics from moving from one place on the hierarchy to another place higher on the hierarchy would be a bottom-up state transition, and vice versa. And so when we do this for a whole series of states sampled from the hierarchy, we have a set of bottom-up and top-down state transitions, and we can subtract those, and then we get an index of the asymmetry in the transition energy that is associated with these state transitions. And this approach kind of allows us to study, whether it's easier or harder to transition in one direction or the other across this gradient of cytoarchitecture. Okay. So when we do this, we find that in general, bottom-up state transitions are easier to complete. So this subplot on the left shows the results we obtain when we apply this approach to a group average connectome from our data set. And this plot demonstrates that bottom-up energy, bottom-up transition energy, so moving dynamics up the hierarchy, is significantly lower than the top-down counterparts. And so this tells us that propagating dynamics from our simple model up the hierarchy is easier than propagating it back down. Most importantly, when we then subtract those energy estimates, so the bottom up and the top down, and we look at the differences, look at the asymmetries, we find that the distance separating brain states across this gradient correlates negatively with this energy asymmetry. So here in the plot in the middle, every point in this scatter plot is a specific state transition. The x-axis is the distance that separates the initial and target state for that transition along the hierarchy. And the y-axis is the difference in transition energies between bottom up and top down. And a negative value on the y-axis means that the bottom up energy is lower than the top down energy. And so what this negative correlation is showing us is that as states get farther and farther apart on that gradient, the energy asymmetry becomes more and more negative, or the bottom up energy becomes progressively easier for the bottom up transitions to be completed compared to the top down counterparts. Then these two plots here in the middle and the far right, these are results for testing that spatial correlation or that distance correlation against a couple of null network models. The details are not so critical here, but really what these nulls tell us is that this correlation we observe is not better explained or not trivially explained by a combination of the spatial embedding of regions in the brain cavity or regions connection weights or strength. So this is effectively saying that this negative correlation with hierarchical distance that we observe is, a consequence of the higher order topology of the connectome, and is not simply born out of the combination of space plus local features. So we think this is a really nice finding because it sort of fits with the idea that regions patterns of feed forward and feedback projections is shaped by sort of similarity and dissimilarity in cytoarchitecture actually may be reflected in the macroscopic level in network topology. So the natural question, of course, is how is that happening? That question is a subject of some ongoing work that we're thinking about at the moment. One simple explanation might be that when activity starts somewhere on the hierarchy and we try to control it to a point that's higher on the hierarchy, that movement of activity or of dynamics is actually routed through a very different set of network paths, or a different set of paths compared to routing that same activity in the opposite direction. If that's the case, then these results would suggest that basically traversing the paths that run bottom up through the brain in our model is easier than top down. Another possibility might be that where activity starts on the gradient may affect how quickly those propagating dynamics hit the densely connected core of the network, and that this might then this might create sort of a multiplying kind of broadcasting effect, which then might affect how easy or difficult it is to complete the rest of the state transition. But these are some points for some ongoing discussions and some ongoing work we're thinking about. Okay, so I think these results are super exciting, but everything I've shown you so far has been based upon the idea of using structural data and using simulated control signals to guide simulated dynamics. And as I touched on before, if these asymmetries in energies are biologically relevant, then we would argue they should be, in theory, be consistent with what we know about observable functional properties that vary over this hierarchy. So in prior literature, for example, from Gao et al., which is shown on the left here, Gao et al. Showed that the timescales of neuronal fluctuation sort of slow down as regions traverse up to the peak of this cytoarchitecture hierarchy, or a proxy of it. And so this plot on the left is showing that regions that are sort of closer to the top of that hierarchy have longer timescales of intrinsic fluctuations measured with ECOG data. And so using their ECOG data, we found some results that kind of complement theirs quite nicely. So this figure on the right is from our own work. Again, every point in this plot here is a state transition like before, but now the x-axis is the change in the length of timescales from that ECOG data between pairs of brain states or patches of cortex. So a positive value on the x-axis now means that as we go from one patch of cortex to another, the intrinsic neuronal fluctuations sort of lengthen or slow down, using that ECOG data. And the y-axis is similar to before, which is the asymmetry between bottom up and top down, with negative being lower bottom up. So this correlation shows us that as, for state transitions where bottom up energy is lower than top down, these state transitions are also characterized by a sort of slowing of neuronal fluctuations or neuronal timescales as we go from one state to another. And so this is consistent with the idea that like a slowing of neuronal fluctuations is, if we think of that as an increasing function of increasing integration, that is occurring higher on the hierarchy. And our results sort of complement this and show that the topology of the structural connectome may actually be optimized to make those transitions more efficient. Okay, so we can validate our results in functional data. That's great. Earlier on, I also said that if these asymmetries were meaningful, they should also show developmental effects. So why did I say that? Well, the reason for this is that the developmental literature tells us that this kind of hierarchical coupling between cytoarchitecture and connectivity is thought to potentially be a program of like a product, of a staged program of development. One of the early phases of this process that is happening during prenatal development is called neurogenesis, which is the process of cells migrating up to the cortex. And neurogenesis is kind of thought to sort of lay the foundation of this for this variation in cytoarchitecture. In particular, just a broad example of that is, during this process, lower order systems that have much more clearly defined cytoarchitecture and clearly defined layers tend to develop later than the higher order systems. They're a bit slower to develop. But then following that, during synaptogenesis, which connecting up those regions, synaptogenesis is then thought to sort of use this blueprint of cytoarchitecture to guide the connection or the formation of connections between regions. And in this case, it seems to flip around, where lower order regions and systems refine their connectivity earlier or first, relative to the higher order systems. So there appears to be a sort of a staging of connectivity refinement that is moving up the hierarchy. Finally, neuroimaging research suggests that this kind of hierarchically staged pattern of connectivity refinement continues well into youth and well into the age range of our sample, which is 8 to 22 years old. So given this, we expected that our energy asymmetries, or our bottom-up and top-down transition energies, should vary systematically as a function of age in our sample. And this figure on the left in the middle shows exactly that. So here what we've done is we've estimated those transition energies between our cytoarchitecture, our patches of cytoarchitecture cortex, separately in each of our subjects. So this is now the subject level rather than a group average connectome. And then what we've done is we've kind of examined how average bottom up and average top down energy sort of varies as a function of age. So this figure shows that both bottom up and top down correlate negatively with age. So the energy is going down as subjects in our sample get older. But you can also see that the top down effect appears to be stronger. And so this kind of suggests to us that these energy asymmetries not only kind of vary systematically throughout youth, but they actually also maybe shrink towards zero, or they close towards zero as a function of age in our sample. Because you recall, I was showing you that there was an asymmetry where bottom up was lower than top down, and now I'm showing you that the top down age effect is stronger. So it's sort of suggesting that the top down is kind of catching up with the bottom up throughout our sample, and they might be converging towards zero. And then this figure on the right shows the age effects for every at a transition level, which are the size of which is shown on the x-axis. And again, the size of just the asymmetry in our group average connectome on the y-axis. And we see a fairly strong negative correlation here. And all this tells us is that, where the age effects are strongest are also where the asymmetries are largest. Again, kind of like converging on this idea that the process of neurodevelopment in our sample is kind of working to close the asymmetry we observe. So this kind of nicely illustrates that like ongoing connectivity refinement, which we think is maybe blueprinted, guided by underlying cytoarchitecture, sort of shapes the energy associated with our transitions, and it sort of is refining throughout neurodevelopment. Okay, so we're almost finishing up. So basically what I've shown you in this middle section is that the topology of the human connectome appears to be sort of wired in such a way to engender asymmetries in the way signals traverse this cortical hierarchy in our model. And we've shown that these asymmetries yield bottom-up transitions being easier than top-down. They seem to be associated with a slowing of neuronal timescales as we move up the hierarchy. And they seem to refine throughout development, which here for us is kind of 8 to 22 years. So far, I've shown you that network control theory can improve our prediction of psychosis. I showed you that at the start, and I've shown you that we can connect our models with some understanding of cytoarchitecture. Now, the final question of the talk I want to turn to is, like, can we take what we've learned from that middle section of connecting our models with cytoarchitecture and feed that back into a prediction framework to kind of improve our prediction of psychosis symptoms even further? So to do that, I just want to very briefly draw your attention to the idea of bystander regions, which are simply the regions that are not a part of an initial or a target state, or one of the two patches of cortex that we're trying to transition between. So for us, that's most of the brain for any given state transition. And a detail that I glossed over about our model is that we implement what's called a full control set. This simply means that all of the nodes of the system are participating in completing a specific state transition, irrespective of where they are in the brain. And they're all allowed this same degree of control. So no regions are given a privileged position to facilitate the state transition. And so what this means is that we know that these bystander regions contribute to the control of state transitions because we let them in the model. And what this gives us an opportunity to study is it allows us to test whether their position along the hierarchy is important for completing, a specific state transition between two patches of cortex. So what we did here, I'm going to keep this very high level, is that we design an approach to imbue our model with knowledge of the hierarchical position of these bystander regions. And so basically this effectively amounted to, giving different bystander regions different amounts of control or privileged positions in the system, depending on their hierarchical position. And then when we did this, we examined how that impacted our asymmetries that we observed in our main effect from before. And importantly, in the subject data, we examined whether doing so improved prediction of psychosis. So just to replicate our main effect, first on the left here, this is just our energy transitions, our energy asymmetry in the group average matrix. And what we find when we take this approach of imbuing our model with cytoarchitecture, that for the bottom up energy is still lower than top down. So this is sort of just reproducing our main effect from before. But the figure on the right is the more important one. Here, what we've shown is each distribution represents the size, the absolute size of the energy asymmetry between bottom up and top down under our uniform, or our standard model, which I was showing for the first part of the talk, which is shown here in blue, and then our control set optimized for cytoarchitecture, which is shown here in the orange color. And the reason we have distributions here is because we reproduce these mean asymmetries under just many bootstraps of our group average connector. And so what this effect shows us, so this figure on the right shows us, is that basically imbuing our model with knowledge of cytoarchitecture actually leads to significant increases in the size of the asymmetry, right? And this is nice because it tells us that, where these bystander regions sit on the hierarchy tells us something about how important they are for facilitating state transitions across the hierarchy. And importantly, the ordering, like adding or imbuing our model with knowledge of this ordering, increases the efficiency of bottom up signaling more than it does top down, which creates like a greater asymmetry. But the really cool thing for us is that we find that this approach improves prediction. So here in this final set of figures, I've come full circle back to the idea of predicting psychosis symptoms from the start of the talk. So here what I've done is I've estimated both like standard transition energy with just a uniform control set, as well as energies optimized for cytoarchitecture, both at the subject level. And then I've used those subject level energy asymmetries to sort of retrain our out of sample regression model that was predicting psychosis symptoms from before. So prediction performance in the orange color in both of these plots is from the optimized control set with cytoarchitecture, while blue is the sort of standard uniform control set. I'm showing a couple of different performance metrics here, but they both tell the same story, which is that optimizing our transition energies with cytoarchitecture yields improved predictions to psychosis symptoms out of sample, which we were really pleased to find. We also find this improvement we also find an improvement in this effect when we're trying to predict age, which is shown on the left, so the same kind of effect. And we also find it when we're trying to predict a measure of executive function, which is kind of a composite score from an online cognitive battery that we acquired in our participants. So clearly, it seems that imbuing our model with knowledge of regions, cytoarchitecture improves our capacity to draw sort of links between the variance in brain network data and people's psychosis symptoms and development and cognition. So there's a question in the chat from Cindy. What about the subcortical structures, participation in the energy differences, since they are not included in the model? Could they be responsible for the higher energy required in the top-down transitions? That's a great question. So that's something that we have omitted here, rather erroneously, of course, is that the connectomes we have are only based on cortical data, so we don't have subcortex in our connectomes, and that's in part because we're trying to study regional properties of cytoarchitecture that we have in the Big Brain Atlas. So I don't have an answer to that question directly, but the thing is that like the question would be like whether including them would close that asymmetry gap. It possibly could. It's something that we haven't tested though, but yeah, it's a great question. Okay, so the answer to my third and final question appears to be yes. It appears that we can optimize our control models to improve the prediction of psychosis symptoms even further by grounding out our models in cytoarchitecture and hopefully improve our understanding of how individual differences in brain networks link to psychosis and other things. So with that, I hope I've kind of convinced you somewhat that we've made some contributions to this goal of kind of leveraging network science to kind of better characterize certain dimensions in this transdiagnostic space. So I just want to finish up by just pointing you towards some resources that we've been developing. So together with some colleagues in the lab, Jason Kim and Jenny Stiso, who are pictured here, the three of us have been putting together a Python package for this control analysis, which is publicly available right now if you follow this link. So it's still very much a work in progress. We still have a lot we want to do, and it's kind of changing all the time. But we wanted to get it out as soon as we could to kind of give people a chance to maybe get in there and have a play with some of the tools and learn about how to do this analysis and maybe do some of the things I showed you today in your own data. Another important part of it is that Jason Kim in particular has put a lot of effort into producing a whole lot of great theory pages that are part of this documentation. And so he's really poured a lot of effort into this, and the hope is to sort of provide you with some intuition on the model, or more intuition than I was able to provide today. So if you're interested in that, we would suggest you check it out, and we're happy to take any questions about it on the GitHub. But for now, I'll just finish up by thanking all of my wonderful mentors, collaborators, and colleagues, etc., who contributed in various ways during my postdoc. Of course, thank you to my funding sources and for everyone for listening. And I look forward to some great questions.
Wow, what a fantastic talk. So clear, really easy to follow. I love the way you kind of managed to connect all those topics. I was wondering if you were going to bring psychosis back at the end, and I like the plot twist. It was really nice. Are there any questions from any people in the audience? Elvisha, nice to see you. Go for it.
Sorry, can you guys hear me? Yep. Hey, great talk, Linden. I really love the entire intro that you had that contextualized everything. I was wondering for the specific transitions or the energies required for the specific transitions, are they stable within an individual over time? Or are they state dependent in any way? So are they affected by sleep, caffeine, alcohol, or in the case of psychosis, like medications or anything like that?
Yeah, it's a good question. So, I mean, recall that this is all based on the structural connectome, right? So it's like, the answer to that question will be yes for the factors that affect variance in the structural connectome, right? So it's like, small state related changes, no. Like if, for example, unless you expect having a coffee at 9 a.m. to change your structural connectome by 11 a.m., then no, the energy wouldn't change. But certainly we would expect like long-term effects of medication could have an impact just as they impact the structural connectome. So yeah, it really just depends on, the answer to that question depends upon what factor you think is changing the connectome, because all of this is just modeling dynamics that aren't like, this is all simulated dynamics, right? So this is not the same as putting someone in an fMRI scanner and saying, well, how does the pattern of observed bold fluctuations change in a state dependent way? We're not looking at that here. So it just depends on what you think changes the structural connectome.
Great, thanks.
Are there other questions in the chat or from anyone? I wouldn't mind going back to Cindy's question, because it's something that I've always wondered. How would you do that? How would you start? Because this is something that's maybe more of a higher level question, but you know, the network neuroscientists are a bit you know, at fault for this, is that, you know, the entire brain really resides in the cortex, which, you know, is a wonderful thought, but it's not true. So how would you connect these subcortical structures into the models like the ones you proposed today?
Yeah, it's a challenge, and you're right. It's something that we sort of often are guilty of burying our heads in the sand about. The reason, one of the challenges is that you know, depending on the edge weight you use for your networks, these models can be very sensitive to like definition of region size. So something that I glossed over is that, for example, we use a parcellation in the work I showed you, the Schaefer parcellation, which is a parcellation based upon functional data. And I tend to like that one because of the size of the regions are kind of relatively homogeneous across, you know, for a given resolution. And then the nice thing about that is that when you build structural connectomes from that parcellation, you end up with fairly consistently sized regions, which plays nicely with structural connectomes. Why am I bringing that up? Well, the reason is, like, if you add in the subcortex, you suddenly can find yourself with either much smaller or much larger regions than the rest of the cortex. And then all of a sudden you'll have control signals that are like dominated by that kind of general difference in region size. It's also, you know, part of the reason why, and this is something reviewers have brought up, like, well, is the reason why I kind of tend to steer away from even using like the Glasser parcellation, which you might argue is, you know, structural is much more appropriate, but the variation in region size kind of makes networks sometimes hard to play with, and that can cause behavior in our models that we see variation in state transitions that are like less, that are, you know, are not quite what we're interested in finding. So there are different ways that people have tried to tackle this, like through normalizing streamline counts by region size, but then we also have found that then changes the dynamics in ways that we don't fully yet understand, and kind of, again, takes the dynamics of our model away from our intuition of what they should be. Again, I don't have stuff to show you for that today, but a bit of a roundabout answer to your question, but the short answer is that it's because it remains kind of difficult, and, you know, we don't just want to throw them in there and then expect it's all good. The actual definition of networks changes in a way that can then make these models a bit more difficult to interpret. And so that's why, like, I remain a bit cautious about it. And so, yeah, the definition of networks is a challenge.
I guess, you know, one thing that I'd be interested in knowing is, you know, I agree that having those subcortical structures in there would be a significant challenge. But what about, you know, doing this in a more targeted way? Like if you're interested in, you know, especially in the context of the psychosis spectrum, like striatal-frontal or striatal-cortical, you know, models, would you see a pathway forward there?
Yeah, sure. I mean, there's no reason why you have to stay at a full connectome scale. So if you have interesting questions about small subcircuits, you certainly can do that. You just have to remember that it's like, you just sort of step away from thinking about specific connectivity and towards thinking about higher levels of topology in the network. So it's a question of like, what do you think? Like, what is it about like corticostriatal circuits, and specifically, like, what is it about topology of corticostriatal circuits that you expect to be, like, say, vary or be different between like cortical projections projecting to the striatum versus striatal projections or paths projecting back to the cortex? It's like, if you have reasons to believe that the topology of those kind of macroscopic circuits is like different, then certainly these models would be cool. But if the asymmetries are kind of if the asymmetries in those predictions are kind of restricted to the same underlying sets of paths, then you would expect like energy asymmetries to be zero. You expect bottom up and top down will be the same.
I think we're at time. So I thank you very much. I also have construction work going on in the background, as you can hear. So I apologize for that. But we're going to shut this down and thank Linden very much for his talk today.
Thank you very much. Thanks for having me. Great fun.